Cell death is a fundamental process in biology. It is essential for maintaining tissue homeostasis, organ development, and protecting against diseases like cancer. Our bodies are constantly making new cells and getting rid of old or damaged ones. This delicate balance is crucial for our health. When this balance is disrupted, it can lead to various diseases. There are two main types of cell death: apoptosis and necrosis. Apoptosis, often called programmed cell death, is a highly regulated and controlled process. It is characterized by distinct morphological changes, such as cell shrinkage, nuclear fragmentation, and DNA cleavage. Necrosis, on the other hand, is an accidental and uncontrolled form of cell death. It usually occurs due to external factors like infection, toxins, or trauma. Apoptosis is like the body's way of removing unwanted or harmful cells without causing inflammation or damage to surrounding cells. Think of it as a controlled demolition of a building. It's a clean and organized process. It is critical for various physiological processes, including embryonic development, immune system regulation, and the elimination of damaged cells. Apoptosis can be triggered by a variety of signals, both from inside and outside the cell. Internal signals can include DNA damage, oxidative stress, or growth factor withdrawal. External signals often come from the immune system, instructing cells that are no longer needed or have become a threat to self-destruct. The intrinsic pathway, also known as the mitochondrial pathway, is activated by internal stress signals within the cell. These signals can include DNA damage, oxidative stress, or growth factor deprivation. When a cell senses these threats, it activates proteins called BH3-only proteins. These proteins promote the release of cytochrome C from the mitochondria into the cytoplasm. Cytochrome C then interacts with other proteins to form a complex called the apoptosome. The apoptosome activates a cascade of enzymes called caspases, which are the executioners of apoptosis. Pause 3S. The granzyme pathway of apoptosis. This pathway involves the introduction of granzyme enzymes into the target cell, leading to the activation of caspases and subsequent cell death. Granzyme B, in particular, is known to activate caspase 3 and caspase 7, which are crucial for the execution phase of apoptosis. This pathway is typically initiated by cytotoxic T cells and natural killer cells. Pause 3S. When these immune cells recognize a target cell, they release perforin, a protein that forms pores in the cell membrane. Through these pores, granzyme enzymes enter the target cell. Once inside, granzyme B directly activates caspases, bypassing the need for upstream signaling pathways. This rapid and efficient method ensures that infected or malignant cells are swiftly eliminated, maintaining the integrity of the organism. Pause 3S. The granzyme pathway exemplifies the body's intricate defense mechanisms, showcasing the precision with which our immune system can target and destroy harmful cells. This pathway not only highlights the elegance of cellular processes, but also underscores the importance of immune surveillance in preventing disease. Section 4. The extrinsic pathway of apoptosis. The extrinsic pathway, also known as the death receptor pathway, is activated by external signals. These signals are typically delivered by specialized immune cells that bind to death receptors on the surface of the target cell. Death receptors are transmembrane proteins that contain a cytoplasmic domain called the death domain. When a death ligand binds to its receptor, it triggers the recruitment of adapter proteins, which then activate caspases. Caspases then carry out the execution phase of apoptosis. Section 5. Necrosis and uncontrolled cell death. Necrosis is a form of cell death that is characterized by its uncontrolled nature. Unlike apoptosis, which is a programmed and organized process, necrosis is typically triggered by acute cellular injury. This injury can be caused by various factors, including trauma, infection, toxins, or loss of blood supply. During necrosis, the cell membrane loses its integrity, leading to the leakage of cellular contents into the surrounding tissue. This leakage triggers an inflammatory response, as the immune system attempts to clear the damaged cells and debris. Necrosis can have detrimental effects on surrounding tissues and organs. Section 6. Conclusion: apoptosis versus necrosis. Apoptosis and necrosis are two distinct processes of cell death. Apoptosis is a programmed and controlled process that is essential for normal development and tissue homeostasis. It is characterized by specific morphological changes, such as cell shrinkage and DNA fragmentation. Necrosis, on the other hand, is an uncontrolled and passive process that occurs in response to acute cellular injury. Understanding the mechanisms of apoptosis and necrosis is crucial for developing therapeutic strategies for various diseases. For instance, inducing apoptosis in cancer cells is a promising approach for cancer treatment, while preventing necrosis in conditions like stroke or heart attack could limit tissue damage and improve patient outcomes.